Hello, my name is Dalton Snyder. I'm a research scientist at Teledyne FLIR, formerly known as FLIR Systems. In this presentation, I'm going to talk about how we can rapidly profile bacterial samples, specifically bacterial response to antibiotics, using two mass spec technologies called Desorption Electrospray ionization and two-dimensional mass spec. And I'll describe what both of those techniques are later in the presentation. But first, I should note that my employer, Teledyne FLIR, makes mass specs and mass spec related equipment and sells those items. And I also have intellectual property related to some of the slides in this presentation. Even so, I've tried to make this as educational as possible. So I'm going to start off by asking the question, what is high throughput screening and why is it important? What is it useful for? So I got this definition from the Encyclopedia of Bioinformatics and Computational Biology. And the definition was high-throughput screening is a high-tech way to hasten the drug discovery process, allowing a quick and efficient screening of large compound libraries. Or you can think of not just screening compounds, but also samples in general. That could be reaction mixtures or biological assays or biological extracts. But screening those types of samples at a rate of a few thousand compounds per day or per week. And specifically for mass spec, typically we think of high throughput as hundreds or thousands of samples screened in a single day. Now, that's not possible with conventional mass spec methodologies. And when I say conventional, I'm thinking about what most labs are set up for, specifically LCMS or liquid chromatography, mass spectrometry. So the liquid chromatography part is where you're separating analytes in your mixture based on their differential interactions with what's called a mobile phase, which is just a solvent, and a stationary phase, which is your chromatography column, which are essentially glass beads. So you send your sample through the chromatography column. The analytes are separated based on their chemistry, and typically your analytes come out one by one in what's called a chromatogram, which you can see in the bottom right of the illustration here. Those analytes are then ionized in the ion source of the mass spec and detected by the mass spec itself. And typically you see protonated molecules if you're using an ion source like Electrospray or atmospheric pressure chemical ionization. So you're measuring the mass of the protonated ion. Instruments typically do what's called MSMS, or a stage of fragmentation, where you're measuring basically the substructure of an ion by fragmenting it in the gas phase. Now, because we're coupling mass spec with chromatography here, our runtimes are typically on the order of 10 to 20 samples, 10 to 20 minutes per sample. If you're doing really deep proteomics or have a really complex sample, perhaps you've got a 1 minute runtime or a 1 hour runtime. And if you're really quick, state of the art, perhaps you've got a 1 minute runtime. But typically you're looking at analysis of tens of samples per hour, which means if you're screening 1 million compounds, that could take 40 years. And that's a long time and obviously not very realistic. So what we aim to do with high throughput screening, or in this particular case, high throughput mass spec, is to screen samples at a rate of thousands to tens of thousands of samples per hour. And we can only do that if the runtime per sample is on the order of seconds. And that means if you do the math, that screening 1 million samples could take just under 2 weeks instead of 40 years, and that's much more realistic. But the only way to do this is to eliminate the slow step, and that means getting rid of the chromatography. Now, when I think of high throughput mass spec, there are a few prominent technologies that come to mind. One is called acoustic ejection mass spec. This is one of the newcomers to high throughput screening. In acoustic ejection, you send a pulse of acoustic energy through your sample solution, which ejects nanoliter-sized droplets from your solution. And so these samples can be in a microplate, for example. you're ejecting nanoliter-sized droplets from those wells into what's called an open port interface, where those droplets are collected and transferred through some diluent solvent to the ion source of your mass spec, where your analytes are ionized for mass spec analysis. A more old school technique, but tried and true, is called matrix-assisted laser desorption ionization, or MALDI, and typically this is coupled with a time of flight or TOF mass analyzer, which is a quick mass analyzer. So in MALDI, typically you're placing your samples on a MALDI plate, a MALDI target plate, and you're depositing an organic matrix on top of each of your samples. Usually that's a small carboxylic acid. You then place this entire sample plate, which has, say, 96 samples on it, into a vacuum chamber where your laser is. You ablate each sample and eject ions from those samples for analysis by mass spec. And so if you either raster the laser or place your sample plate on an XY stage that can be rastered, then you could analyze samples very rapidly. The technique I'm going to talk about today is called Desorption Electrospray Ionization, or DESI, which we refer to as an ambient ionization technique. And what that means is that ions are generated from a sample at atmospheric pressure in the ambient environment with little to no sample preparation. That's in contrast to MALDI, which takes place in vacuum. So desorption electrospray ionization involves directing a spray of charged droplets onto a dried sample. That solvent creates a thin film on the sample spot. Further impacts of primary microdroplets then eject secondary microdroplets containing your analytes of interest, which have been extracted into that solvent. Those secondary droplets proceed into the inlet of your mass spec, where they can be mass analyzed. So how do you do high throughput sample analysis with this DESI mass spec technique? Typically, you're starting off with all of your samples in some sort of master plate. That can be a 96 well plate, a 384 well plate, a deep well block, for example. And your samples might consist of organic reaction mixtures, where you're varying the concentrations of each of the reagents, or you're varying the groups on each of your reagents, or you're varying the pH, etc. They can also be biological assays, where you're testing a biological response to an antibiotic or to an inhibitor or something like that, or where you're testing various time points or pHs and so forth. In any case, you transfer all those solutions from your master plate to what we call a DESI sample plate, which you can see in this white rectangular box here. You can do that with a variety of tools. One is a pin tool. So this example shows a 384 pin tool, where we're transferring 50 nanoliter droplets to a DESI sample plate. That's usually a Teflon coated substrate. So you let those droplets that are transferred onto the Teflon dry. You transfer that sample plate to the front of your mass spec where the ion source is, and you raster through your samples to analyze each one of them one by one. And Purdue, which is right down the road here in West Lafayette, has a state-of-the-art high-throughput DESI mass spec system inspired by the DARPA Make It program, where basically they were trying to analyze as many samples as possible, as quickly as possible. So again, you could be looking at organic reaction mixtures or biological assays, or biological extracts, for example. So extracts of bacteria in our case. You've got all of these in some kind of well block. You transfer those solutions from your well block to your DESI plate, which again is a Teflon coated substrate. You let those droplets dry, leaving your dried analytes on the DESI sample plate. You place the sample plate in front of your mass spec where the ion source is, and you raster the samples through the ion source region to analyze each sample successively. Now, what kinds of tools are needed to do this high throughput DESI mass spec analysis? Well, if we're automating this entire process, then we need some sort of liquid handling robot, or a 96 channel pipette, for example, or a multi-channel pipette. You might need some additional robotics. So for example, a robot arm to transfer sample plates from your liquid handling robot to the front of a mass spec. You also need your mass spec, which has your DESI ion source and XY stage. That can be any kind of mass spec, from a TOF to an ion trap. In this work, we're typically using an ion trap mass spec. And then, of course, you need to do some data processing with all this data that's coming out of your mass spec. And that's usually done on a cluster or a single computer, something like that. And in this work, we're interested in doing high throughput analysis of bacterial samples, specifically looking at the response of bacteria to antibiotics by DESI mass spec. So Purdue has previously demonstrated this experiment wherein they're able to monitor bacterial growth over time. In this case, it was E. coli, and you can see in the plot on the right-hand side, this is an optical density at 600 nanometers measurement, where the x-axis is hours and the vertical axis is the optical density. And so you can measure that the bacteria is growing and multiplying. By mass spec, we can do something called two-dimensional mass spec, or 2DMS, which generates a mass spectrum that has two dimensions of mass to charge. The horizontal dimension is your precursor mass to charge, or your intact mass. These are protonated analytes. And your vertical dimension you can think of as an MS2 dimension, or as a fragmentation dimension. The analytes shown here are from a lipid profile. That is their PE lipids, phosphatidylethanolamines, and PG lipids, phosphatidylglycerols. And you can see they offer some shared fragmentation routes. So if you draw a diagonal line through all the PEs, you can see that they have similar neutral losses. And if you draw a diagonal line through some of the PGs, you can see they have similar neutral losses. And we'll get into that in a little bit more detail later. But if we use this technique to look at how the E. coli is growing over time, we can see in the plot on the right-hand side that initially there are smaller and simpler lipids that are observed, but as the E. coli is growing, we see not only larger and more complex lipids, but a more complex lipidome overall. Now, I've broken this experiment down into 4 core technologies that you need to understand in order to do this experiment. So what I'm not going to talk about is the mass spectrometer itself. It's just a generic ion trap mass spec. I am going to talk about the front end of the mass spec, or the ion source, because a key part of this experiment is the high throughput desorption electrospray ion source with your XY stage. There's also this thing called 2D mass spec analysis, which I talked a little bit about on the last slide, but I'll discuss this in greater detail in a bit, where we can acquire these really unique mass spectral profiles that look like images, because they are essentially images, for different bacteria species and for bacteria species exposed to antibiotics. You can see a couple interesting examples here. Obviously, if we're talking about laboratory automation, we're interested in sample prep robotics, sample handling robotics, so liquid handling robots, multi-channel pipettes, that sort of thing. And then finally, no high throughput workflow would be complete without some sort of high throughput data analysis pipeline. And for us, looking at the metabolome and lipidome of different bacteria species, that's a bioinformatics pipeline. And it'll also show you that AI and machine learning can be useful for processing our data as well. The first core technology was a DESI ion source. So Eric at Purdue and Graham Cook's group has this really nice paper in IGMS where he shows what it takes to do this whole DESI-MS experiment with an XY stage. So DESI involves the spray of charged droplets onto your sample. Your sample is extracted into a thin film of solvent, and then further impacts of primary microdroplets produce secondary droplets that go into your mass spec for analysis. So you need your nebulizing gas coming into your DESI sprayer, you need solvent coming in, and you need some, I think these are swage lock fittings that make up this DESI sprayer. Your samples are typically deposited on some sort of DESI plate that's either a microscope slide or a Teflon coated substrate. That's what this is, a Teflon coated glass slide. Usually when you're doing DESI, you're utilizing a kind of a janky ion transfer tube where it extends out from the front of the instrument so that it can reach not just the DESI ion source, but all the samples on your sample plate. And then, of course, you've got the mass spec itself. Now, your sample plate is sitting on this 2D moving stage, an XY stage. And of course, that has its own electronics associated with it, specifically stepper motors or piezoelectric motors, and some sort of driver for those stepper motors. And of course, you can buy all-in-one XY stage solutions that come with the motors and the controller. This little video on the right-hand side sort of shows you the DESI process here. So the ion source, the DESI ion source, and the mass spec inlet are stationary. And the way you go from sample to sample is by rastering your XY stage. So you can see we're going from sample to sample and analyzing each spot successively in time. Now, hardware is only half the battle. There's also software to consider. So our collaborators at Purdue use a MATLAB-based approach where they've got mass spec control, data and data post-processing integrated into this MATLAB GUI. They can also control their XY stage by using Excel-based movement files, so telling the stage how quickly to move, where to move, that sort of thing, what sort of pattern to move in once you're on a sample spot. At FLIR, we use a Python-based approach for our R&D. So on the top right here, you see our mass spec software that has lots of real-time and post-processing options. And then on the bottom, we've got our XY stage control software. So telling the instrument which samples we'd like to analyze, how quickly we'd like to raster through them, what kind of rastering we want to do, how many mass scans we want for each sample. You can also integrate some data visualization tools here. So for example, this color indicates the intensity of this mass to charge 743, or it could indicate the overall intensity of the mass spectrum on that particular spot. Second technology I wanted to talk about is called two-dimensional mass spec, or 2DMS or 2D MSMS. Those are all the same thing. So to understand this technique, you need to know what the 2D MSMS data domain is. So the horizontal axis in this top plot in the top right, you can think of as an MS1 dimension. Basically, those are your intact precursor ion masses, or the molecular weights of your compounds. And then the vertical axis, you can think of as an MS2 dimension. So a fragmentation pattern for every precursor ion. Now, if you're familiar with triple quadrupole mass specs, then you're familiar with things like product ion scans, precursor ion scans, and neutral loss scans, which are useful for analyzing complex mixtures for similar functional groups. Usually you think you can only do this on triple quad instruments, but we showed at Purdue that you can do these on an ion trap as well, which is somewhat surprising. And you can do even fancy versions of precursor and neutral loss scans in the top right here on an ion trap. But once we figured out we could do that, actually this was my graduate work, we figured out we could actually get the entire 2DMS spectrum, that is acquire an MS1 spectrum and MS2 spectra for every precursor ion in the same scan. We initially did this by some fancy frequency encoding technique, but the data was difficult to interpret and hard to calibrate. So Lucas, also in Graham Cook's group, figured out how to do this in a way where the data generated was both interpretable and calibratable. And so we can generate an entire 2D mass spectrum now with an ion trap using a single scan. So let me show you what a 2D mass spectrum looks like and tell you how to interpret it. Typically, when you look at a mass spectrum, you've got one dimension of mass, which you can see in this plot on the right-hand side. There's one dimension of mass to charge and one dimension of abundance, which is the vertical dimension. Now, measuring a single mass or mass to charge only gives you a tentative ID for a molecule because you can have isobars, isomers, various collections of your elements and atoms that could create an ion with a particular mass, and we only have so much mass accuracy and resolution. So typically, we increase the accuracy of our identifications by using something called MSMS or MS2 or tandem mass spec. And the way we usually do it is by isolating each compound one at a time and fragmenting them to generate an MS2 fragmentation pattern. In 2D mass spec, we do this all with a single scan. So we generate a true two-dimensional mass spectrum where we measure the masses of all your precursor ions and generate a fragmentation spectrum for every precursor ion. And if we apply this to bacterial species, we find that we can generate really unique lipid fingerprints for each species. This is an example of four sort of model organisms that we've been looking at in our lab: E. coli, Pseudomonas aeruginosa, Staph aureus, and Bacillus subtilis. And you can see each of these 2D profiles shown on the bottom is really unique. For example, E. coli is predominantly PE lipids or phosphatidylethanolamines. Pseudomonas aeruginosa consists of not just PEs, but also phosphatidylglycerols and diacylglycerols and fatty acids. From Staph aureus, we detect lysyl PGs, diglucosyl diacylglycerols, as well as DAGs. And in B. subtilis, we see this unique set of cyclic peptides at high mass to charge. The third core technology, part of what makes this high throughput, is the sample handling robotics. And this ranges in capability, level automation, and cost from simple tools like pin tools and manual multi-channel pipettes up to complete liquid handling robots. These are your Teakins, your Hamiltons, your Beckman Biomex, those sorts of things. And there are even devices in the middle, like 96-channel pipettes that are fully automated and smaller liquid handling robots. And the things you can do with liquid handling robots are automate essentially all of your biological assays. So what we do, for example, is we can translate an exposure response experiment where we're treating a bacteria with an antibiotic to see its response to that antibiotic. We can translate that to a deep well block format and automate the entire process, including the growth of the biological material, the exposure, of course, and the sample collection. We can also automate these CFU bioassays to see which samples died and which are still alive. And then, of course, we can automate the preparation of the samples for analysis either by high throughput DESI mass spec or through liquid chromatography mass spec, which you see in the bottom right there. And if you've got a great team of engineers supporting you, you can do all sorts of things with these robots. For example, we've developed custom labware adapters where we can use these robots with HPLC vials, with Eppendorf tubes, with microscope slides, all sorts of different shapes and sizes of labware. And we can also automate our DESI plate prep. So, for example, we can transfer each sample to a small Teflon-coated microscope slide, which you see in the middle picture here. Or you can do a more large-scale transfer with a 96-channel pipette, as shown in the picture on the right-hand side. These plates are much larger. They're the size of a 96-well plate, essentially. After you've transferred your liquid samples to these Teflon-coated substrates, you let them dry, of course, and then you transfer them to the front of your mass spec, where you can do your high-throughput analysis. And of course, we're generating a lot of data per unit of time, so we need a way of processing that data and generating useful information from that data. So, you've got to think, what questions am I trying to answer with my current experiment, and how do I answer those questions by extracting useful data? So the Chopra Group, another one of our collaborators at Purdue, has this pipeline they call CLAW for analyzing LCMS data, specifically MRM data, where we can very quickly annotate lipids, for example. So we can say which peaks correspond to which lipids. We can do statistical analyses if we have technical and biological replicates. And then we can do some molecular networking. So If we know that certain species are upregulated or downregulated, what processes and what other molecules are those targets related to? But of course, AI is all the rage these days, and we found that AI and machine learning are great tools for processing 2D mass spec data. Um, for example, I've got a set of 4 bacteria spectra. These are lipid spectra of B. subtilis, B. thuringiensis, Staph aureus, and S. capitis. And as a human, using your neural network, your brain, you could tell these spectra apart. And so can a neural network if you train it properly. It's like training a human to identify a dog or a cat or a bird. If you see enough pictures of a dog or a cat or a bird, you can accurately identify them using your neural network. So that's what we've done here is we've collected a lot of data from a set of 8 bacteria, and we've trained certain AI models to identify those bacteria based on their lipid profiles. And the results from that are shown on this next slide. So we have 4 models that we've tested: a random forest classifier, a k-nearest neighbors classifier, a multi-layer perceptron classifier, and my favorite, a convolutional neural network, which you can think of as an image analysis tool. And all of these yield fairly high accuracy, nearing 100%, but the most promising method we found was the convolutional neural network. This is a really unique way of analyzing mass spec data because typically your mass spec data is not in the form of an image. The unique thing about 2D mass spec data is that every mass spectrum is an image that can be used to train a convolutional neural network for identification purposes. So we think this is a really promising avenue. Now, in the end, what do we want to do with this high throughput DESI 2DMS system? Well, we want to profile bacterial response to antibiotics. So if I've got E. coli and I expose it to, say, gentamicin, what are the metabolomic or lipidomic markers that indicate whether the antibiotic has been effective or not? So we expose these bacteria to antibiotics in a high throughput manner on a liquid handling robot, and then we profile these samples very rapidly with DESI 2DMS, and then we compare that data to LCMS data that we're collecting in parallel. So that's the experiment I just described. We're automating our exposure response protocols. So we expose our bacteria to various concentrations of antibiotics. We do some sample processing and sample handling and prepare those samples for analysis by DESI 2DMS or LCMS. We compare those data to each other, and we also do statistical analysis of that data and to generate some data visualization plots so that we can interpret the data. And eventually, we'd also like to use AI machine learning tools in this pipeline, but we're not quite there yet. And here's the system that we're using. It's a prototype system for doing DESI 2DMS. It's an ion trap based mass spec that we've been working on for the last year. You can see on the right-hand side, it's got a DESI ion source on it with a DESI XY stage, which is the big black thing on the bottom there. And then the mass spec sits behind the stage. And this is the kind of data we get from this prototype system. In this experiment, we exposed Pseudomonas aeruginosa, which is a model bacterial system, to some antibiotic. We also had a control experiment that was not exposed to the antibiotic. And we collected these 2D lipid profiles using the DESI 2DMS system. And what's really important here is the difference between the control and the antibiotic spectra. You can see that there's a greater abundance of these lyso-PE lipids when the organism is exposed to this antibiotic. And so we take that as a chemical signature of that the antibiotic has been effective in this case. And we can do this profiling very rapidly on the order of hundreds or thousands of samples per hour. Now, typically, when you start going high throughput, you give a little bit up in terms of the analysis fidelity, i.e. your data is not quite as good as it was when you were doing sort of the gold standard slower technique. So I have a comparison here between your gold standard LCMS-MS that we've done in the lab and our DESI 2DMS system. All these peaks labeled in green are peaks that are shared between the two analyses, and all the peaks in red, which are, in this case, mostly PC lipids, phosphatidylcholines, were not detected above the noise, at least, on the DESI system. Even so, we're able to detect 60 to 70% typically of the top 30 compounds with the DESI system, analyzing samples at a rate of hundreds of samples per hour rather than 3 samples per hour on our LCMS system, since our runtimes are around 20 minutes per sample. Now, no technique is without its limitations, so I wanted to talk about those for a minute. The advantages of a high-throughput DESI 2DMS system are pretty obvious. One, the sample throughput is obviously very high compared to chromatography. You're looking at analyzing hundreds, if not thousands, of samples per hour versus a typical chromatography run is 10 to 20 minutes. So you're looking at maybe 3, 4, 5 samples per hour. Mass spec is known as a high sensitivity technique, but also has high molecular specificity because we're doing a stage of MSMS. It's also widely applicable depending on what kind of ion source you're using. And of course, all of our MSMS data from the 2DMS experiment can be compared with existing data in all those MSMS libraries out there. The challenges of a DESI 2DMS system are that, like mass spec in general, it's an expensive technique. It certainly requires users to have significant knowledge and lab skills. We think that's a problem that is solvable, though, through some clever engineering. It does require hours-long culture times, and we prefer not to wait an hour to analyze our samples. It is easy to overload or contaminate a mass spec, and because a mass spec is widely applicable, depending on the ion source, you can also have high chemical noise, so high background. And then our mass spec happens to be an ion trap, and ion traps are known for having limited intrasample dynamic range, but that's a solvable problem if you switch to like a TOF analyzer, for example, which has a greater dynamic range. Even so, I think the advantages of the 2DMS system far outweigh the challenges. And that's all I had, so thank you for listening to this talk.